Okay. First of all, my name is Rawan Obeid. I work in the Palestinian Ministry of Health in the National Center for Cancer Diagnostics and Human Genetics. I have a bachelor's degree in biotechnology and a master's degree in molecular genetics and genetic toxicology. Today, my topic is going to be about targeted panels and using the next generation sequencing technology. And then I will be discussing a case study in which this technology was used in identifying a novel mutation. The topics I will be going through today are summarized in this agenda. First, I will be discussing going through some points, what is DNA sequencing, and comparing it to other techniques. Then I'll be going through the solid tumor targeted panels and their importance. Later on, I will discuss the methodology and how this technique works. Then I'll be presenting my case study that signifies the importance of NGS. Okay, so what is DNA sequencing? This is a background that I think most of us have. So sequencing means to determine the order of the first four chemical building blocks, which are the bases that we all know, the A, C, T, and G, that make up the DNA molecule. Why do we use DNA sequencing? So in medicine, it's used for a lot of purposes, diagnosis and treatment of disease. We can know if the gene has a mutation that is related to a phenotype of a disease. Obviously, now there is a wide range of uses for sequencing. It does not only limit itself to clinical use. Okay, so what's the difference between Sanger sequencing and next generation sequencing? Because one technique is not better than the other, rather than each technique has its own usage. So the critical difference between Sanger sequencing and the NGS is sequencing volume. So the Sanger method only sequences a single DNA fragment at a time, while the Sanger method sequences massively parallel, sequencing millions of fragments simultaneously per run. So NGS offers superior sensitivity compared to Sanger sequencing, particularly in detecting low frequency variants and rare mutations. Also, sequencing has more depth, and it can identify subtle genetic variations present at frequencies almost as low as 1%. Okay, Sanger is more cost effective, but for smaller targets, but NGS is more cost effective for higher targets, more targets than one gene. For turnaround time, the Sanger sequencing is obviously more rapid since it only sequences some genes, little as 30 minutes per, for example, a small fragment from 250 to 300 bases, while NGS, it's 3 hours, but 3 hours is actually lying to you because 3 hours is how much the platform takes to sequence, while the wet work takes a day and a half before actually putting it on the sequencing platform. Okay, so here I'm going to talk about the usage. So which cases should we use Sanger and in which cases should we use the next generation sequencing? I'm just going to give you an example for the familial Mediterranean fever disease. So we have a known, the gene that causes familial Mediterranean fever is the MEFV gene. It has a known genotype. For example, we know known and reported pathogenic variants on Cosmic, on ClinVar, that they are found in exon 2, 3, and 10, some of which are found in exon 5 of this gene. So we sequence this gene. It takes up to 2 hours almost to sequence these exons only that are known to have pathogenic variants and analyze their results. Sorry. However, in next generation sequencing, we're not going to use it for a single disease with a known genotype. We can use it for cancers, autism, hereditary diseases with high levels of phenotypic heterogeneity. And we can also use the Sanger to validate the results of the NGS. Okay, what are the uses of the next generation sequencing? We can use, of course, it depends on the platform, but almost all platforms, we can use genome sequencing, exome sequencing, and targeted panel sequencing. Genome sequencing, as you all know, it targets all genes coding and non-coding, so exons and introns of the gene. However, the depth is lower since it covers a large area. It has a very long turnaround time, and the accuracy is a bit low. For exome sequencing, it's going to cover the entire exome from 20 to 25,000 genes. The accuracy is better. It's more cost effective, and the depth can reach 100x. The targeted gene panel, which today I'm going to be discussing, it only covers gene sets from 10 to almost 500 genes, according to how the manufacturer designed this gene panel. It can cover up to 500x in depth, and it's the most cost effective, and it has a very rapid turnaround time. However, I just want to comment on something here. This is rapid turnaround time, and the whole exome says long turnaround time. Now we have platforms, for example, we do whole exome sequencing on the Ion Torrent platform, which does not have a long turnaround time. It's actually also 4 hours per sequencing the chip, and it's also a day and a half of wet work. So the time now to exome sequence almost takes the same time to do a targeted gene panel. And there's a fourth use that I didn't mention here, which is RNA sequencing as well. Okay, so the solid tumor targeted panels, these are examples that I have obtained from manufacturers as an example of the targeted panels that are already designed. Of course, you have some, I think, like the Thermo Fisher, they allow you to design your own panel. For example, if you are interested in some genes or you know a certain cancer that has certain genes, you can design your own panel. But here you have already designed panels for human lung cancer panel, for example, and the human actionable solid tumor panel. These are the genes that it covers. And here it mentions, for example, which cancer contains which genes. Which mutations are found in which genes, obviously. Okay, so what's the importance of targeted panels? The importance lies in the targeted therapy. So we all know that the medicine now advanced, and we don't just use chemotherapy for all cancers. There are many targeted therapies out there in the market. Basically, some targeted therapies, patients develop resistance to these targeted therapies according to different mutations they obtain on the receptors. So we can know all of these information according to with the usage of targeted panels on the sequencing platform. This is just an example here of the importance. So this is non-small cell lung carcinoma example. So these are the main targets, which are 8 targets of the targeted therapy. Okay, so here, if, for example, the patient's EGFR positive, which occurs in almost 15% of non-small cell lung carcinoma, that's 1 in every 7 cases. So this is very important to know if the patient has the EGFR mutation or not. So we can use the tyrosine kinase inhibitors correctly and with better prognosis. So here we have these 8 target genes, and these genes have developed, for example, a patient has developed targeted therapy resistance by having activation mutation of the EGFR gene with these mutations, amplification of the MET, fusion of the ALK. So these are important to know with the usage of targeted panels for better patient survival, prognosis, and to enhance the targeted therapy for each patient. Another case here is the importance of targeted panels for an example, in the case of colorectal cancer in BRAF gene. So BRAF, as we know, up to 15% of colorectal cancer cases have mutations in the BRAF gene. Of those mutations is valine 602 glutamic acid. This accounts to 99% of the mutations that happen in the BRAF gene. So what's the importance in this case to know that this mutation is there for this particular patient? Usually for people with CRC, the treatment is cetuximab plus chemotherapy. Okay, so in this case, if the patient harbors this mutation, the patient will not respond to the standard treatment. So in this case, or in this study, they found that using these three, or the triplet targeted drugs, or the doublet of the encorafenib and binimetinib, this has improved overall survival and tumor response rate relative to those who received only the standard treatment, which is the cetuximab plus chemotherapy. Okay. So what is the workflow of targeted panels? This is the main workflow, and then I will go to details. We extract the nucleic acid that we have according to the tissue or the blood sample we have. Then we do a library preparation. The library preparation involves, but is not limited to, fragmentation and then adapter ligation to give a unique sequence to each patient, sequencing the library using whatever platform you have, and then the analysis of the data. When the sequencer generates the FASTA-Q files, we align the reads into a BAM file, and then we use the VCF file to identify the variants or analyze our patient data. Okay, so in this case, when we, as I mentioned, I'm going to be discussing solid tumor panels. We have a biopsy. Usually the sample is a biopsy, and then that biopsy is embedded in formalin-fixed paraffin. And then this FFPE tissue is used. It comes to the lab, and then we take scrolls from it. And then these scrolls, we have to obviously remove the paraffin and just take the tissue itself. So the paraffin removal is usually done by a deparaffinizer or xylene according to the protocol you're using in your lab. The sample is lysed. There's DNA cross-linking step to remove the artifacts or the impurities from DNA. And then we use the spin column for DNA binding. We wash the DNA, and then we elute it to have a product to use for NGS and other genomic applications for pipeline validation and assay development. Okay. So there are many types of sequencing. I think my colleague discussed it in the first presentation, and she discussed the various types of sequencing. But in this situation, I'm going to be discussing the Solexa sequencing that we use for the targeted panels. So Solexa sequencing uses the library preparation. We use fragmentation, enzymatic fragmentation, and then these fragments are cut into blunt ends. The blunt ends, we ligate adapters to them. The adapters are called capture sequences. The adapter part on the outer part, they bind to the flow cell. We need them to bind to the oligonucleotides that are already, or the binding sites that are already found on the flow cell. That is provided by the company, of course. And then the inner sequence is used for the primer ligation and the polymerase ligation to continue or sequence each target. So the library is hybridized onto the flow cell, and then we have a bridge amplification cycles to amplify the clusters, because obviously in sequencing, you need a next generation sequencing. You don't need one signal like we do in Sanger sequencing, which is only one. Here we need hundreds of thousands of signals, so we can obtain, and we know that this is a true variant, and we can obtain a very strong signal from the sequencer. So we have the amplified clusters here after the amplification. It also reads forward, and then it reads the reverse. After the clusters are amplified, as I told you, in the inner sequence, the primer binds and the sequencing cycles start. And it uses here the same concept as Sanger sequencing, which you know is by DDNTPs, or deoxyribonucleic acids, dideoxyribonucleic acids, which have the hydroxyl group removed. So it prevents the addition of another DNTP after it. Basically, they are fluorescently labeled nucleotides, and each has a color that signifies which nucleotide. For example, here the C is blue, T is green, and then this is the data collection, and this is what the flow cell looks like. Usually, it's lighting up like lights. And then after the data collection is collected on the flow cell, alignment and data analysis follows. The read clusters, they align with overlapping regions, and then the sequences assemble by because we have multiple patients, obviously, in the same primer pool and in the same pool and in the same library. We have to, or the alignment tools help us to each patient is to segregate each patient عن الثاني. Okay. Next. Okay, so here I'll be discussing the case study that I mentioned earlier. How could we use this platform and the targeted panels to identify novel mutations? So this is one of the vital uses of the Sanger the NGS that I mentioned. This was a study that I made in the Arab American University. It's a functional assessment of a novel mutation in the SMAD4 gene in a Palestinian cancer patient that has CRC or colorectal cancer. So why is studying cancer in the targeted panels important? Because the worldwide statistics for cancer morbidity and mortality obtained from the WHO, and it was released in 2020. These two bar graphs, as you can see, have the colorectal cancer and the lung cancer as the second and third highest cases worldwide, and the first and second most common cancer death, respectively. In Palestine, these are the Palestinian Ministry of Health statistics. In 2022, also mirror the WHO findings, where colorectal cancer comes in second place, lung cancer comes in third place of the top most common cancers, which also are the top two cancers that cause the morbidity and that cause the mortality in the Palestinian population. What were the aims of this study? It's to contribute to the growing body of knowledge on the genetic landscape of colorectal cancer, and it employs scientifically advanced techniques to identify novel mutations in the Palestinian colorectal cancer patient. It provides insights to the molecular mechanisms underlying the disease, and it paves the way for targeted therapies tailored to the Palestinian population, which I think is the most vital here. How was the study designed? It's basically how I mentioned before, which with a few other additions. We prepare the DNA sample by extracting it also from FFPE tissue. And then we sequence it on the sequencing platform that we have. And then the FASTA-Q files that were generated, we use the patient's clinical information to facilitate the variant analysis for clinical diagnosis using oncology interpretation software. However, sometimes the targeted panels that are designed by the manufacturer gives you no pathogenic variants or no likely pathogenic variants found, which are the pathogenic variants, of course, that are reported. So what to do then? Is the patient negative? Does the patient have no variants? No. Here is where the study comes in handy. So we took the VCF file and we removed the filters, and we took all of the variants that are of unknown significance. Okay, and then we did in silico analysis to predict their pathogenicity, and if the patient has anything that can contribute to the metastasis or to the development of cancer. And then after we saw candidate mutations and segregated them by in silico analysis, we did validation of these analyses by Sanger sequencing. And then since the mutation that I'll be discussing now that I found was found to be a pathogenic variant, SMAD4 plays a vital role in gene transcription, and that this mutation can cause gene loss of function. So I designed the experiment in order to have a functional assay that determines the mutation's effect on gene transcription. So I found that the most beneficial functional assay to do here is luciferase assay. So the luciferase assay here, we had plasmids that are sent in bacteria as agar stabs. The plasmids that I used have already incorporated SMAD4 in them. And then I introduced my mutation by site-directed mutagenesis using the kit Quick Change Lightning. I inserted my mutation, which is SMAD4 position 115 cysteine to arginine, into the following plasmids, which are these two plasmids. And then I had both wild type and mutant. And then the nicked vector DNA containing the desired mutation, I transformed it into XL10 gold ultra-competent cells. These cells, after cloning them, they were maxi-prepped to get higher purity and a higher number of plasmids, which were then, I wanted to use them for transfection or cell transfection. Before cell transfection, I seeded HEC293 T cells. After 48 hours of transfection, 48 hours of seeding, the cells were transfected with both the wild type and the mutant plasmids, and they were co-transfected with the luciferase reporter plasmid, so we can have fluorescence and we can report it. Later, the luminometer was used to detect the transcription levels of the gene. So the dual assay, luciferase reporter assay, was used. First, we lysed the cell, and then we measured the lysate's activity. Okay, so the results here in the in silico analysis that I showed you, when I told you that the targeted panels did not give me any positive pathogenic or likely pathogenic variants, and these are the VUSs generated, I used various in silico tools, SIFT analysis, polyphen analysis, Perovian, aligned GVGD, and then the conservation was found based on confirmed using clustral omega, because if the area is not conserved, we can't consider it as a true variant, as you know. And then also the amino acid change was studied. So after all of these analyses were done, SMAD4 position 115 cysteine to arginine was the one I chose to continue with the functional analysis of, because as you can see here, all of them gave me that it's very deleterious and likely damaging and likely to affect protein function. Based on these preliminary in silico results, I decided that it's the most deleterious, and I visualized the mutation using protein 3D structure, which I'll be showing you shortly. Why is SMAD4 important? I studied it to understand how it can affect the CRC cancer. Here, SMAD4 is the co-SMAD in the TGF-beta signaling pathway. This signaling pathway promotes many physiological processes, including cell growth, differentiation, proliferation, fibrosis, and scar formation. So if anything goes wrong in this pathway, it's going to drive metastasis, prevent apoptosis, and so on and so forth. So in the SMAD4, this mutation, if I lose the SMAD4, which is the co-SMAD, which allows this complex, the SMAD2 and the SMAD3, to enter the cell nucleus and go on with the gene transcription. If I lose, loss of SMAD4 will not allow the SMAD complex to enter the nucleus for gene expression, and therefore I will be losing the gene expression of many genes. Okay. Here, non-synonymous SMAD4 mutations reported across various tumor types from CBioPortal and Cosmic are illustrated in these bar graphs. CBioPortal graphs shows that SMAD4 mutations are mostly present in pancreatic, colorectal cancer, lung adenocarcinoma, and this result is mirrored in Cosmic as pancreatic, large intestine, biliary tract. So the mutations are widely spread in these types of cancers. So here I know that I am still validating that this mutation has something to do with my patient that has colorectal cancer. This photo showing SMAD4 missense mutations in colorectal cancer distributed along SMAD4 domains was obtained from CBioPortal. SMAD proteins have two evolutionary conserved regions. We have to understand, obviously, when we want to study mutations, we have to understand on which domains it lies, so we can understand the significance of this mutation. So in this case, I have two main domains, the MAD homology 1 and the MAD homology 2. They're separated by a linker region. The N-terminal MH1 is responsible for sequence-specific DNA binding. MAD homology 2 is responsible for heteromerization and transactivation functions. So, in conclusion, or we can conclude here that any mutation that happens in the MAD homology 1 will be responsible or will alter the DNA binding of this gene. So my mutation is here. It's on the MAD homology 1. And then many studies results demonstrate that the MAD homology 1 domain as a whole is very sensitive to changes. So that tumorigenic mutations within the region leucine 43 to arginine 135 indeed would target DNA binding. And as you can see, even though this is a novel mutation, it lies within the region that a previous study mentioned that any changes to this region will affect DNA binding. So here, 3D protein structure visualization will help us understand more how will this mutation affect DNA binding and how deleterious it is. So the changes here, you have the zinc. It is bound to C71, C115, which is mine, C127, and histidine 132. This zinc finger critically stabilizes the packaging of this MAD homology 1 motif and the conformation of the DNA binding hairpin. Okay, so consequently, SMAD protein is predicted to be functionally dead if this mutation happens. So why did I do segregation and validation of SMAD4 mutation by Sanger sequencing? As I told you before, NGS variants or variants detected by NGS technology can be validated by Sanger sequencing in order to confirm the results. And there's another reason for it, to know if the mutation is somatic or germline. So we took the peripheral blood of the patient and the mother and any family members that we have, and we found that the mutations are negative in these electrographs. They show negative results for the mutation. It's in the coding region 343 T to C. So it's still T here. So this confirms that the mutation is not familial and is somatic. So this also signifies that the heterogeneity of cancer and that it happened while while the division of the cancer was happening. Hello? Okay. Anyway, to assess the effect of SMAD4 mutation on the transcriptional activity of SMAD4 gene, we used luciferase assay. In this situation, luciferase assay, these results show the tested mutation, which is SMAD4 position 115 cysteine to arginine, decreased the SMAD4 transcriptional activity by 4-folds. As you can see here, it's the wild type SMAD4. It's at 1. The mutant SMAD4, they were obviously normalized against the empty vector or the backbone. The wild type SMAD4 was, it's at 1, and the mutant SMAD4 is at 0.25. So the mutant SMAD4 or the mutation that I have inserted decreased the SMAD4 transcriptional activity by 4-folds. And here, as you can see, the p-value is less than 0.001, which is very significant. Okay, so what is the conclusion? Next generation sequencing has revolutionized our ability to identify unique mutation and has enhanced our understanding of cancer biology. This knowledge can provide a landscape of cancer genomics in Palestine. So it's very crucial to understand the underlying genetics of cancer. Why? For it can also be personalized for not only the Palestinian community. I'm just using this as an example because we live in the Palestinian community, which does not have a lot of studies covering this topic. This could be personalized for each and every community, for example, each country. Now, this is important because when we create a database, I can understand, is this a true variant? Or is this found in the Palestinian community as a normal mutation? Again, I can also use personalized therapies for specific communities. For example, this variant is specific and found in the Palestinian community or found in the UK or found in the US. So this is crucial for understanding the unique genetic factors that contribute to cancer in diverse populations. Thank you for listening, and I will take any questions now.
شكرا لك أخت روان، يعطيك العافية.
الله يعافيك، شكرا.
أي حدا عنده سؤال أو استفسار من الزملاء؟ Any questions for روان؟
Okay.